Minesh-shaytanir-rajim. Bismillahir-rahmanir-rahim. Lamajmu'un ila miqati yawmin ma'lum. Summa innakum ajwazaluna al-muqazzibun. Ila akiluna min shajarin min zakum. Famali'una min halbatun. Fasharibuna alayhi min al-hamim. Fasharibuna shurb al-him. Hadha nuzuluhum yawm ad-din. Nahnu khalaqnakum falawla tusaddiqun. Afara'aytum ma tumnun. Antum takhluqunahu. Antum takhluqunahu. Am nahnu al-khaliqun. Sadaqallahu al-aliyyul Azim. Number 45 for 9th class with a topic, with a brand new topic. The topic which I just want to discuss today is centrioles. Centrioles. You are watching the spelling in the blue color, CENTRIOLES, the centrioles. That is visible on the screen. Okay, G. It is present in the animal cell and this is absent in the plant cell. Centrioles are present in the animal cell and they are absent in the plant cells. And what is the major function of this organelle? Because I just started from the cell wall, I was discussing other organelles. You see the importance of these organelles. Cell wall, primary cell wall composed of cellulose, secondary cell wall composed of lignin. Function, protection of the cell, just like the skeletal system in the human body. Cell membrane, lipid protein composition in the cell membrane. Look at these organelles. Look at the importance of these organelles. Cell membrane, a hero in the sense that allows some molecules to pass into it, and some molecules cannot enter into the cell because that is the semi-permeable membrane. We go back to the nucleus. That is the in charge, the boss of the cell, the main boss of the cell, which consists of chromosomes, which consists of the DNA, and the DNA is a hereditary material. Chromosome composed of two things, DNA and the protein. What is the name of the protein? Do you remember? Histone. And DNA is hereditary material. What is the definition of hereditary material? I am recalling previous sections. I am recalling the previous sections before discussing the centrioles. Cell membrane is semi-permeable membrane. The DNA is hereditary material. Why it is hereditary material? Why DNA is more important? Because DNA transmits the character from the parent to the offspring. Mean that any character which you possess, that is coming from the father and that is coming through the DNA, and DNA consists of 4 important molecules which I will teach you in the senior sessions. That sequence of these molecules in the DNA are so important because each character, each disease is being handled out by these structures. DNA, come back to centrioles, another organelle. Of course, the Golgi complex I have to tell you. Of course, I have to tell you some transport vesicle, lysosomes. You will listen in the upcoming sessions. Now the centrioles, where they are present? They are present in the animal cell. In which cell they are not present? In the plant cell. They consist of membrane or they do not consist of the membrane? This is a question. This is a very precious question, important question, that the centrioles consist of membrane or they do not consist of membrane. Answer is they consist of membrane. Mean they are membranous organelle. I will write here the membranous organelle. They consist of a membrane. I am just going to name very significant protein which is present in the centriole. But I will tell you, first of all, the membranous organelle and the non-membranous organelle. We were discussing in the last sessions. Tell me, the centrioles will be present in which cell? Just recall the previous knowledge and just tell me that the centrioles will be present in which cell? In prokaryotic cell or eukaryotic cell? Yes, they will be present in eukaryotic cell. The reason is that because they consist of the membrane. The membranous organelle present in the eukaryotes and non-membranous organelles are present in the prokaryote. You want me to define the definition of the prokaryote, eukaryote? I did that earlier also. I will do this again and again. The well-defined nucleus present, that cell is called the eukaryote. The well-defined nucleus absent, that is called the prokaryote. Now what is the definition of the well-defined? Well-defined mean nuclear membrane present, that is the well-defined nucleus. When the nuclear membrane is absent, that means that well-defined nucleus not present, mean DNA present, chromosome present, just a nuclear membrane is not present. Now you are confused, I know, because when you were studying the structure of the nucleus, you were studying the nuclear envelope that was surrounding the nucleolus and the chromosomes. Of course, nucleolus and the chromosomes are surrounded by the membrane called nuclear envelope. So I was naming nuclear membrane, you was confused. So I am clarifying. Nuclear membrane, nuclear envelopes, they are the both same things. So don't worry about that. That is the same thing. Nuclear envelope, nuclear membrane, they are the name of the same membrane. We'll go back to the centrioles. How many cylinders, I am using the word reason because they have cylindrical shape. Then I will name technically as well. I will draw one cylinder here. Look here, this is the one cylinder. This is the one cylinder which I am making here, and that is another cylinder. This is the another cylinder, and these two cylinders I have made. These two cylinders collectively called centrioles. These are the centrioles. One cylinder is called the centrioles. One cylinder is called centrioles. Correction. One cylinder is called centrioles. Both cylinders collectively will be called as a word which I am just highlighting you. That is called as a centrosome. Now question is, sir, you was telling that nucleus consists of hereditary material. Question is, sir, you was talking about the nucleus, that nucleus is composed of three types of RNA, transfer RNA, messenger RNA, ribosomal RNA. Question is, sir, messenger RNA is very important. That is getting the message from the DNA was going to the ribosome by endoplasmic reticulum. Question is, endoplasmic reticulum was transporting the molecules from the nucleus to the cytoplasm, from the cytoplasm to the nucleus. Question is, ribosomes were forming the proteins. Question is that the ribosomes function to form the protein in the formation of the protein, mean protein synthesis. And question is that the ribosomes were having the two units, large ribosomal unit, small ribosomal unit. Question is, when these two units combine, the protein formation starts. Question is that these two units will combine when messenger RNA will come after getting the message from the DNA, from the nucleus towards the ribosomes, and then ribosomes will form the protein. Question is that the chromosome consists of DNA and the protein. Question is that cell membrane allows some molecules to pass through it and refuse some molecules to enter into the cell. Question is, cell wall is much protective structure for the plant cell. What is the function of the centrioles? Golgi complex and remaining functions I will tell you later on. Sir, what is the question of the centrioles? That is $1 million question. Centrioles. You will see, first of all, in which cell they are present? In the animal cell. In which cell they are absent? In the plant cell. Now you will see, I was talking about the centrioles. They are involved. You have to write now because that is much important information I am telling you. That is a much precious information I am telling you. And that information will tell you why the centrioles are present in the animal cell. These information will tell you why they are present in eukaryotic cell. My question once again to you, which organelle present in the prokaryotic cell? Ribosomes. You are right. Why ribosomes are present in the prokaryote and ribosomes are present in eukaryote as well? Because the ribosomes are without membrane. Yes, any non-membranous organelle, ribosomes, where they are present in both cells. If they are present in both cells, is there any difference between the ribosomes of the eukaryotic cell and the ribosomes of the prokaryotic cell? Yes, there is a difference. Yes, there is a difference because you have to, nature has made the different distinction between the ribosomes of the prokaryote and the ribosomes of the eukaryote. What was the distinction? Distinction is that the ribosome of the prokaryote is the smaller in the size and the ribosome of the eukaryote is the larger in the size. That is the distinction. The centrioles. One centriole here, one cylinder, and that is the another cylinder. This is the another cylinder. Two centrioles collectively called as centrosome. Now I will teach you one centriole internal structure. One centriole internal structure. This is the front part of the centriole. This is the front part of the centriole. I want your attention because when you will see microscopically, because they are visible by the microscope, they are not visible by naked eye. When you will see by microscope, you will find 9 tubes. 1, 2, 3, 4, 5, 6, 7, 8 and 9. The 9 tubes are present in one cylinder. 9 tubes are present in the one cylinder. You are watching the 9 tubes. I am not talking about the both cylinders. I am talking about the one cylinder. In one cylinder, the 9 tubes are present, and these 9 tubes, each tube is further divided into 3 tubes. So which word you will use? The 9 triplet microtubules. The word I have used, the microtubule, that is important word, and that's the word I was discussing in the cytoskeleton. What is a microtubule? That is a polymer. That is a polymer. What is a polymer? Large molecule is called the polymer. What is the name of the small molecule? The small molecule is called the monomer. What is the relationship between the polymer and the monomer? Small molecules combine to form the large molecule. Monomers combine to form the large molecule. Polymer and water molecule will be released in condensational polymerization. So microtubule, the word the microtubule. What is the meaning of the micro? That is the protein. That is the protein, but it is polymer, mean many protein molecules will combine to form the large molecule, mean polymer. And what is the monomer in this case? Tubulin. So students, the tubulin, which is the monomer, which is the protein, which is the smaller molecule, it will combine to form the larger molecule, that is the polymer. You are watching. Reactant I have written, the product I have written, red color indicator indicated that the tubulin changes to protein, and the process is called condensational polymerization, in which you are converting the small molecule into the large molecule. So centriole composed of a polymer. What is the name of the polymer? Microtubule. What is the name of the monomer? Tubulin. T-U-B-U-L-I-N. The tubulin. What is the name of the large molecule? Polymer. Protein. What is the name of the small molecule? Tubulin. The tubulin combined to form the large molecule. Name the large molecule polymer. So that is the way to form the polymer. I go back to the centriole structure. Centriole structure consists of how many tubes? You forgot? I will recall once again the 9 tubes. 1, 2, 3. You are just confused about the cylinder. This is the cylinder. This is the cylinder. I am talking about the one cylinder, mean one centriole. It consists of the 9 tubes. 1, 2, 3, 4, 5, 6, 7, 8, 9. The 9 tubes. And each tube further consists of 3. So which technical word you will write in the paper? 9 triplet microtubule. You are telling me something? You are telling that total number of the tubes are 27. You are right. You are technically you are right, mathematically you are right, because 9 multiplied by 3 equal to 27. Because 9 triplet mean, I will also show this figure to you. By this figure you will be able to better understand. Look here. Look here, the actual figure came with the 3 different colors. You can see that the cross section of the 9 triplet microtubules. Now I will count one by one with the different colors. Okay, you can see this is the number 1, this is the number 2. This is the number 3, 4, 5, 6, 7, 8, 9. The 9 triplet. How many tubes are mentioned in the figure? 3. The total number of 9 triplet microtubules are present in this one centriole. So what is the number of the tubes in the 2 centrioles? In one animal cell, 2 centrioles are present. Still you want the answer that what is the function of centriole. I am going to tell you. First function I have already told you. The microtubules are the part of cytoskeleton. They give the protection to the cell, because the cell is without skeletal system. Your body consists of bones, but the centrioles are without any skeletal system. So Allah Ta'ala made some fibers inside the cell, means cytoskeleton, cyto means cytoplasm. To protect the cell, some significant structures were being awarded to the cell, and one of them is centriole. But major function is the cell division. Yes, cell division. Cell division means that during the cell division, centrioles play significant role. They become 4. I will not teach you this point later on, because in the cell division chapter we will discuss in much detail. Here I will tell you only the one sentence. The centrioles help in the formation of the cell division. So I am talking about the 9 triplet microtubules, centriole structure. What is the function of centriole? What is the function of centrioles? They help in the cell division. Centriole present in which cell? They are present in the animal cell. They are absent in the plant cell. How many centrioles present in one animal cell? 2. Where it is present? In the cytoplasm. In the cytoplasm. What is the function of the centrioles? To protect the cell, help in the cell division. To help in the cell division. How many tubes are present? 9 tubes are present. 9 tubes are present. 9 tubes of the centrioles are present. If you will see here, I will make the tubes once again. 9 tubes are present. So 9 tubes are present. Look here, one tube, second tube, third tube, fourth one, fifth one, sixth one, seventh one, eighth one, ninth one. So 9 triplet microtubules are present. 9 triplet microtubules are present. So if you will see this one, I am just going to tell you the 9 triplet microtubules are present, and these 9 triplet microtubules in one centrioles and 9 triplet microtubules in the other centrioles, and these tubules will tell us that these tubules will explain how they help in the cell division. Now you will see, I will draw 2 centrioles once again, and I will use that color which is visible to you. This is the one centriole, one cylinder you can see here, and now I will make another centriole. This is another centriole. What is the name of the structure which is formed by the combination of the 2 centrioles? Mean when 2 centrioles will combine, what is the name of the structure? Centrosome. 2 centrioles collectively called as a centrosome. How many centrosomes are present in one animal cell? 1. How many centrioles present in one animal cell? 2. Why I am telling you these number of the centrioles? Because you will be little confused when I will tell you number of the centrioles during the cell division. Cell divisions you will study in much detail, but one point I will tell you, you are growing day by day. Your size is being increased, your hairs are being increased, nails increased, your height increased, your body become little fat sometime when you are eating more. Mean number of the cells are increasing. When the number of the cells will increase, that is due to the cell division. I am not naming that cell divisions now. Okay, I name mitosis and meiosis, but I will not name here much detail because we will go in much detail in the next session. So mitosis increase the number of the cells, mean you will grow. How you will grow? The centrioles will be involved, the chromosomes will be involved. You study the chromosome in the nucleus, you studied the chromosomes in the nucleus. So centrioles are indispensable, they must be present in the cell, otherwise the cell will not grow. Your questions are in the plant cell, they are not present. How the plant cells are growing? How the plant stem is increasing in the size? Because cell division they also need, because they are also living thing, they are also growing. I will tell you the reason. They consist of microtubules. They do not consist of the centrioles, but they consist of the microtubule. What is the composition of the centrioles? Microtubule. So actually centrioles are composed of microtubule. Once again, how many tubes are present? Because I am focusing one point, the structure of the centrioles. The 9 tube, 1 tube, second tube, third tube, fourth one, fifth one, sixth one, seventh one, eighth one, ninth one. And each tube, when I will enlarge this one, when I will enlarge this circle, each tube further consists of how many tubes? 3 tubes. In each tube, 3 tubes. In each tube, 3 tubes. Which word you will use to justify this statement? 9 triplet microtubules. Now my last point of today, that centrioles when they will divide, the centriole, 2 centrioles normally present. I told you number of the centrioles in the animal cell. When animal cell is not dividing, when it is not dividing, they are 2 in numbers. When they will divide, they will change into 4. They will change into 4. Now point, this is much important point. This is much important point. I will repeat that centrioles normally they are 2, but when they will divide, the cell will divide, they will change into 4. 2 centriole at one place of the cell and 2 centrioles at the other place of the cells. Between them fibers will be formed. These fibers, new words are coming, because you know that when I will use these words again and again, you will be used to, because I am using one term so many times that they will go into blood and mind. So don't worry about the words. The word I am using, I will be slow in speaking this word. The word is spindle fiber. The word is the spindle fiber. So what is happening? The centrioles, you can see the 2 centrioles in one direction and other centriole in one direction. Sir, 2 centrioles should be present because you are making the 4. 2 here you are making, 2 here you are making. Technically animal cell consists of 2 centrioles. Now you are making 4 centrioles. Sir, this is a contradiction in the statements, because earlier you was telling animal consists of 2 centrioles. Now you are making the 4 centrioles. Sir, what is the reason? The reason is that during cell division, 2 centrioles divide to form the 4 centrioles. So what is the statement? What is the statement? I am issuing a statement. During the cell division, animal cell consists of 4 centrioles. In the normal cell, when the cell is not dividing, that consists of 2 centrioles. What is the term you will use when 2 centrioles will collectively? Which term you will say? Centrosome. Mathematically, one centrosome consists of how many centrioles? 2. How many total number of the tubes present in one centrioles? 9. How many total number of the tubes collectively mathematically present in one centriole? 9 triplet microtubule. If you will not use the word triplet, total number of the tubes in one centriole, 9 multiplied by 3 equal to 27. Total number of the microtubules in the 2 centrioles, 27 multiplied by 2, 54. During the cell division, total number of the microtubules in the cell, because they will change it from 2 to 4, 54 multiplied by 2, 5108. That is the mathematical data for the centriole. So important organelle. I am unable to say that which organelle is not important, because when I was talking about nucleus, I was telling that is a boss. When I was talking about the cell wall, I told that is a protective system. When I was talking about the cell membrane, bilipid layer and the protein and the fluid mosaic model, those are much important, I was telling you. Similarly, each organelle, Golgi complex, I will tell you, I already recorded those lectures about the Golgi complex. You will see very soon, and you will see that is just like a factory. You are making a biscuits in a factory, and you are exporting, delivering inside the country or outside the country. I will explain. You will enjoy that lecture. So my sons, centrioles, some information I have given you, and I will continue. This was lecture number 45. Thank you very much. Allah Hafiz.